Ok.
Sí, ya.
¿Sí?
Sí, sí.
Bueno, pues buenos días. Hoy bueno, voy a hacer la presentación en español porque para presentar a Juan quiero hacerla, quiero hacerla con tranquilidad. Entonces, de verdad es un verdadero placer y un honor presentar a una persona y a un científico del nivel de Juan Pérez Mercader, como le gusta decir a él, que investiga en la frontera entre la física, la biología y la exploración planetaria. Y sobre todo para muchos de vosotros y muchos de nosotros ya lo conocemos desde hace bastante, pero sobre todo para las nuevas generaciones que tenemos en el centro y los nuevos que no lo conozcan, creo que es importante resaltar la figura de Juan. Nació en Alcalá de Guadaira, en Sevilla, y aunque luego fue criado en Huelva, pero vamos, andaluz de pura cepa. Hizo y estudió ciencias físicas en la Universidad de Sevilla. Después tuvo una estancia en el Trinity College en Dublín. Tras ganar una beca Fulbright, desplazó a Nueva York, donde hizo un máster y se doctoró en física teórica, en el City College de Nueva York y en la Universidad de Nueva York. Regresó más tarde a España y se incorporó al Consejo Superior de Investigaciones Científicas, donde bueno, llegó hasta profesor de investigación, con un montón de trabajos y de investigaciones, y mantenía el carácter de científico consultant en el Laboratorio Nacional de los Álamos, además de profesor externo en el Instituto de Santa Fe, en Nuevo México. Su gran inquietud e interés por las ciencias multitransdisciplinares pues le llevó a promover, junto con otros científicos, el estudio de la astrobiología y en concreto todo esto lo plasmó en la creación y la fundación del Centro de Astrobiología aquí en Madrid, y es digamos, nada más y nada menos una de las hitos y de las digamos, más importantes entendido de su carrera, porque además gracias a ese empeño personal que él puso, por supuesto, consiguió el apoyo de todas las instituciones, que ya es complicado en este país, y se creó el centro mixto del Centro de Astrobiología CSIC-INTA, ya allá por 1998, y que se plasmó en noviembre de 1999 con el acuerdo entre las dos instituciones. Fue el director del CAP hasta 2008, donde bueno, pues su inquietud también investigadora le movió pues quería seguir haciendo cosas, quería plasmar muchas de las ideas que él tenía en la cabeza, que le habían rondado durante todo ese tiempo, y quería pues seguir trabajando, y bueno, pues lo que pues se desplazó a Harvard, donde se incorporó al departamento de ciencias planetarias, y donde pues está haciendo una gran labor, pues con el mismo entusiasmo de siempre, la misma pasión y una capacidad de trabajo, la verdad que impresionante. Pues está haciendo una gran labor que nos contará seguidamente en sus investigaciones. Juan ha trabajado, como digo, es multifacético y en múltiples aspectos de la física teórica, de partículas elementales, cosmología, astrofísica, materia condensada, física de no equilibrio. Ha hecho, como veremos ahora, sus pinitos en la biología, en la teoría relatividad, general de la relatividad, en fin, la química experimental, y por supuesto toda la contribución que ha hecho al desarrollo de instrumentación también para la exploración planetaria. Ha recibido numerosas condecoraciones, premios, en fin, que no vamos a entrar a detallar porque nos tiene que dar el seminario. Pero bueno, y hoy nos va a hablar sobre todo de uno de los trabajos más importantes que está desarrollando en su laboratorio, que es cómo hacer vida sintética, como yo creo, vida sintética de verdad, es decir, dejando a un lado los componentes biológicos y bioquímicos, y él nos los va a contar seguidamente. Han hecho de verdad contribuciones muy relevantes que han sido meritorias de portadas de revistas tan importantes como Angewandte Chemie y bueno, en fin, le dejo que él ya nos cuente sus trabajos y sin más, Juan, agradecerte de nuevo por estar aquí con nosotros, aquí en tu casa, por madrugar un poquito, aunque ya sabemos que para ti eso no es madrugar mucho, pero bueno, muchas gracias y te escuchamos con impaciencia.
Buenos días, Víctor. Buenos días, queridos todos, queridos todos con arroba, queridas amigas y queridos amigos y personas que no conozco. Me supongo que el seminario es en inglés o es en castellano, no lo sé, o no recuerdo. Víctor,
en principio es en inglés, pero como te parezca que bien, está bien.
Not a problem. Good morning, everybody. It's a pleasure to be back here, and it's a pleasure to be talking to CAB, Centro de Astrobiología, or Center for Astrobiology. It's a pleasure because, as Victor has remarked, I have something to do with it, but also it's a pleasure because I see that it's growing. I see that work which is of relevance, scientific work and technical work which is of relevance is beginning to be recognized everywhere, and that's something which I really enjoy seeing. And although I'm not participating in that, but I enjoy seeing it. Today I will be talking about what it says there: mimicking simple life in the laboratory without using biochemistry. So what I will do is I'll give a brief introduction about why we do this. I will also then talk a little bit about what features of life do we want to mimic and why. And then I will try and tell you how we do it and what we have achieved so far. And so without any further ado, I would like to start. But before starting the actual talk, I would like to say something which I consider is important. It's a few words remembering our colleague, Carmen Morales Duran, who died a couple of days ago. And so Carmen Mamen, together with others, and in particular, when we started live, Albert and I found a colleague there, other colleagues as well, but today is Mamen's day. And we just became very good friends. We not only became very good friends, we also began to do things together and so on and so forth in science. And we learned a couple of days ago through Benjamin, myself, through Benjamin, that Benjamin Montesinos, that Carmen had passed away. So it generated sadness in me, of course, great sadness. And I would like to, I mean, this is not a medium to actually have a little moment, but I would like to ask people in the audience to please keep 5 seconds, 10 seconds of remembrance of our colleague Mamen. So Mamen, rest in peace. We appreciate you, we loved you, and we miss you. Thank you, everybody. So after this necessarily sad moment, let me start talking about the science. So we know that all terran life, all life that is that we know so far, which is life on Earth, is based on standard physical-chemical processes. There is no vital force or any thing of that kind. It's standard physical chemistry, except that, of course, it does it with biochemistry. All living systems, from bacteria to ourselves or the oak trees outside this house and so on, are out of equilibrium physical chemical systems, and I insist in physical chemical systems, which are controlled and implemented, as I just mentioned, by biochemistry. But if you look at today, life on Earth, it looks really complicated, but it is also complex and systematically organized. But we do know that life on Earth has been here for about 3.6, 3.7, maybe 3.8 billion years ago, American billion years ago. And the planet is 4.567 billion years old. So did the jump in complexity occur suddenly, or did the jump of complexity occur more gradually? Well, there is a huge leap between the chemistry that probably was organized in our planet at the time, or also chemistry that may have come from the interstellar medium that was here, and the presence of living systems. So there is, as I say there in red, there are huge leaps in complexity from chemistry itself to biochemistry with lipids and so on and so forth, and then get it organized into life. So in many phenomena that we see and study in physics, for example, the structure of the universe, for example, the world of subatomic particles everywhere, we see that the complexity that apparently occurs at the largest scales is a reflection of very deep principles which are very simple. So there is a simplicity somewhere hiding at some level, at some scale. This is, of course, something which is extremely fruitful, or has been extremely fruitful in cosmology, in particle physics, in condensed matter physics, in many areas. Can one extend this line of thought to biology or to life? Does one need biology? Well, just thinking about this is very imposing. It's very imposing because the complexity is really appalling. If you look, for example, at a simple bacteria, some object like Escherichia coli, which is between here to here is about 2.5 microns, and the diameter is about half a micron. You look at that, and then you look in this. This is, of course, this picture here. If you look at that in more detail, you become really astounded. If you look at, you see the bacterium. This is the cellular wall. This is what it looks like in electronic microscopy. And my colleague in the top floor of my building, Howard Berg, and others have been investigating, for example, and have had an extremely fruitful scientific life, investigating, for example, how the flagella of the bacterium move. And they have discovered in incredible amounts of detail that the bacterium is actually, the flagella is actually powered by a turbo engine made with proteins. And here is the actual conformation of the proteins and how the flagellum works and so on and so forth. So how complex can this be? I mean, it's extremely complex, but of course, this had a lot of time to develop from maybe simpler forms of life. Today, we know very well that all living systems that we know in the Earth have a common ancestor, LUCA, which stands for Last Universal Common Ancestor. This is about 3.8 or so billion years ago. And at this time, at LUCA, where the eukaryotes, the archaea, and the bacteria seem to have a common trunk, okay? At this point, there already was biochemistry with DNA, RNA, etc. So, of course, there must be a huge jump around here, around this time, but from the beginning of the planet. So, could there be is there a period of time where simpler systems have been evolving that eventually came to be a basis for the archaea, for the bacteria, and then eventually split kept evolving, and found our friend Luca here, and then it became life as we know it now today, with the three major domains. This is called the Protocell Era, around this time here to here. So could there be some simpler form which still didn't use, this probably may have begun to use DNA, RNA, etc. But could there be something even simpler? Well, people like our friend and colleague, our friend here, who won the Carrefour Prize a few days ago, or a few weeks ago, and likes to say, can we look for, this is Luca, and then can we look for forebear lineages from before the most recent common ancestor, Luca? And how complicated were these guys? Well, this is what we are going to be trying to do. See if we can find some way to approach this era in a way which is illustrated or founded in the way physicists think. We know that life in our planet has been evolving for billions of years, okay, the 3.8 or 3.6, 3.7. And we also know that it is quite complex and also quite robust. It is, reason tells us that because we do see today that things go from simpler to more complex, is the arrow of time, is the growth in disorder, the tendency to grow in disorder, which requires more complexity to deal with it. So reason tells us that at early stages, it probably was far simpler. So the question that a physicist like myself with huge interest in understanding this problem, the question that I ask is, what is the simplest mimic we can synthesize in a one-pot reaction to remind us of the little warm pond or warm little pond that our friend Darwin used to ponder about, pun intended, between pond and ponder? What is the simplest life mimic, that is to say chemical system, that we can put together, that we can synthesize in one pot using small molecules, meaning by that not using any biochemistry? So for that, to tackle that problem, we can think of bringing the methods and power of physics in the sense of unification, and try to unify all these phenomena that we know about life, all these properties that we know about life, using the tools of the scientific method today. Today, the scientific method involves, as I say down there, and I am almost reading, involves a combination about mathematical representation, which we can use as a guiding principle, just as Newton used a guiding principle for to actually unify gravitation as the cause for the Earth rotating around the sun and the planets rotating around the sun, and also for apples falling on the Earth. That type of unification is a consequence of the mathematical representation. Of course, you need a Newton, but a mathematical representation. Today, we also use computation as a means of using with the mathematical representation, or inspired or aided by the mathematical representation, we use computation. And finally, the test of the pudding is in the tasting, which is we have to do experiments to confirm whatever we see, or experiments to inform whatever we see. And the experiments, the computation, and the mathematical representation finally need to be informed by a good understanding of the phenomena we're dealing with. So this is what we're going to be talking in today's talk. And here is a very short summary of what you will see after this introduction. You will see a little bit of theory and things it predicts. Then we will see some theory-directed experiments in synthetic non-biochemical polymer chemistry. We will introduce what affectionately is called PISA, polymerization-induced self-assembly. And then I will show to you, I'll describe to you how building an autonomous, self-booting, totally synthetic, non-biochemical protocell using in the form of a supramolecular object in the 1 to 10 microns size range, and using one single pot a homogeneous reactor, a topoietic system, okay, how we do that and where we are. And I will also talk a little bit about work, which is right now being reviewed, submitted a couple of weeks ago, and work which is on my desk, and work which is in the laboratory. I will describe some of that. And so let's go, and then I will offer a few conclusions. So there is a very important point that we are standing on the shoulders of giants. Darwin is somebody who already in the book, The Origin of Species, he began to talk about what today we consider are the basic properties of life, and which is where I am heading. I'm going to just make a brief introduction, but I also want to use history so as to put things in perspective about what are the properties which are shared by living systems. And Darwin already, the properties, of course, are the handling of information, the capacity to metabolize, the capacity to self-reproduce, and the capacity to evolve using adaptive and non-adaptive strategies. The adaptive strategies go all the way back to Charles Darwin and others. But Charles Darwin didn't think about information. Information was first began to be discussed in the history of this huge intellectual enterprise in which we are involved, understanding how to make life or how life occurred here. It didn't get discussed until Hermann Müller began to realize the role of genes in evolution, after, of course, the work of the Abbott. Then a few years, this is in the 20s, and then a few years later, Schrödinger, a major figure in quantum mechanics, a major figure in physics in the 1940s, when he was in Ireland, he gave a series of lectures trying to bring physical thinking, or the thinking of physics, into biology. And he, of course, produced this extraordinary little book, What is Life?, which was read by everybody, and that led to a new generation of molecular biologists and so on. And the discoveries that occurred in the late 40s and then the early 50s are a consequence, and into the 60s are a direct consequence, people say, of Schrödinger's book and Schrödinger's work. After that, there have been people who have been thinking about this unification of the forces, this unification of the properties of living systems and how to implement them. And most notably amongst them is Tibor Ganti, who put together an idea with a mathematical description, which goes under the name of the chemoton, which he published in Biosystems in 1975. But that is a very brief, totally lopsided, or very partisan description of this evolution of the intellectual history. It's totally subjective. But what are the properties? The properties that characterize living systems are, as I mentioned very briefly earlier, are that the systems handle information in the same way in which you make a computation, where information is input, information is transformed, information is output, and then information can also be changed. Systems do that. That's what is meant by saying handling information. With that information, the system is capable of, and its chemistry, the chemistry of the system is capable of capturing molecules, and its simpler molecules in general, in principle, in its environment, and actually being able to actually make more molecules of the ones which are needed to keep the system going. And eventually the system, there are some processes going on that make the system replicate as a way of keeping the species up, and which can in some sense be understood as a way in which life fights, in quote, fights against the second law of thermodynamics. What it does, of course, the second law of thermodynamics is unavoidable, and living systems are open systems, so they are not themselves subject to the second law, but then under environment R. Okay, so then systems, living systems, are capable of doing self-reproduction or self-replication of the system in a way which is programmed and controlled by the living system itself. And finally, the living system is capable of evolving, and is capable of evolving, adapting to the environment, or not, or simply surviving to changes in the environment or changes within the system itself. This is called non-adaptive evolution. An example of that is a mutation. The system is capable, if the mutation is not very super strong or anything like that, the system is capable of surviving, mending, or adapting, or surviving in the environment in which it is. So living systems do all of that, and examples of that. This is a transparency taken from a book, undergraduate book. Examples of that can be seen in living systems. Notice that I keep saying self-replication, self-reproduction. That means that viruses which depend on a host to actually self-replicate are not being considered in what I am discussing today. Okay, so the living system unifies all these four properties, and it's non-equilibrium chemistry, which is also very important, and it's shared by all known living systems today. All of them are unified into what we call the cell division cycle. Here you have an abstract view of the cell division cycle. This is extracted from a beautiful book by Andy Murray and Tim Hunt, which was published in 1993, and where they depict here in a cartoon how the cell division cycle occurs. In the left-hand side, y-axis, you have the cell mass, which is associated with metabolism, roughly. And on the right-hand side, the y-axis, you have the DNA per nucleus, which can be assimilated in some way to the amount of information in the system. The system starts with, notice that the Y scale here has the 1 here. The Y scale in the left-hand Y-axis has the 1 in a particular place, which is different than this one. These are in arbitrary units. So the way this is represented here, you have the system starts with 1 unit of mass. The system starts with 1 unit of information in DNA, and then the mass of the system as time begins to evolve, to flow, the mass begins to grow, and eventually reaches a particular time around here, where the mass is twice the mass of the original system. The information stays constant for a while, and then after a little bit, there is a beginning of the DNA replication process in which the system goes from unit 1 to unit 2, and then it stays there during a time while the mass keeps growing, and then there is chromosome segregation here, and a little bit later, the system actually divides and replicates. It's called the cell, and then it repeats. The children, the daughters replicate, and so on and so forth. And then there are all kinds of strategies to do this, from sexual strategies to many other sports and so on. So there is this unification of all the properties of life, which is actually represented in the cell division cycle. So if you are like myself, a physicist, then you would say, what is the spherical cow? So, you know, the simplest imagination that the simplest system you could imagine about how to describe life. So what will be involved in a simple mathematical, physical, chemical representation of the properties I just mentioned? Well, you will need chemistry, of course. And in chemistry, you need a mechanism to react and another one to transport the molecules. So a simple way of thinking of that is by imagining a reaction and diffusion system where the molecules are involved in a reaction and they diffuse. This, of course, this type of systems were introduced by Alan Turing in the early 50s. You will need at least two substances, but 3 actually. you do need two substances, one, an imaginary substance, a meta chemical substance, which would represent the cytoplasm, the cell itself. And we would call that V. And another metachemical substance, which will represent the food that the system is taking from the outside. That food, which the system is taking from the outside, we will represent that metachemical species, we will represent by U. But then the chemical systems, living systems, degrade, produce products that are degraded as a consequence of metabolism and the processes that take place inside the system, and also the interactions with the environment, and they produce some waste, C. So you will need at least these three substances, V, U, and C, in addition to the reaction and diffusion of the molecules. You will also want to represent the environment. Living systems do not exist in a void. Living systems exist in an environment. And the environment contains a random intrinsic component. For example, in the feed, or for example, in the temperatures, the chemistry is affected by noise, and you will also need a noise in the system. To represent these properties, information, you will need a linear and quadratic function in these substances to actually represent on-off switch, from which you can do together with linear operations with an on-off switch, which is highly nonlinear. You can do any mathematical operation. So you will need a kinetics that contains terms of this type, algebraically of this type. You would need metabolism, and for that you could think of representing it with autocatalysis terms in the reaction system, like U squared, V squared, or UV. You would need self-replication, and for that you could think of Turing patterns, for which you need a cubic interaction term, like V cubed, UV squared, etc. And then, in order to have evolution, it is well known from people who study control and adaptive behavior, you will need two feedback loops, including operation in the random media. How do you put all of this together? Well, the way to put all of that together is to actually make polynomials like this and then apply the mass action kinetics. And here is what you think, what you get. You get a system where you have the three substances, you have the interior, and in the interior, you can think of it as a lattice, the interior of the living system. You can think of it as a lattice. The lattice contains in each of the nodes of the lattice, that could be some kinetic, there is in the proposal we have, there is a kinetics which represents what I just mentioned, and it is constrained by mass action kinetics. And you come up with equations like this, which we started thinking about this in life with only, unfortunately, with only two substances. And then later on here, we have developed it further to represent what you will see in a minute, a system with three reaction diffusion equations, where this is the diffusion terms. This is the various components that I described earlier. This is this eta U, eta V, eta C represents the noise, etc. And when you put that, and that system with this diffusion terms actually are capable of representing adaptation just by those terms. And when you put that into a computer, this is now in review in Physics of Life. It was submitted 2 weeks ago. And but we have been working on this for years. And so the system looks like this, and so on and so forth. You can prove that there is a very rich variety of morphologies that are generated by the solutions to these equations. And you begin to see that there are systems, there are areas. This is the parameter space for the equations. And there are areas there where there is cell-like behavior, which is predicted. And if you put that in a computer, you begin to see that the system is actually capable of generating a system of equations generating that. If you look at it in some more detail, you see that you start with an initial condition, which is you put some perturbation in the medium, some perturbation of V in the medium, and then the system begins to grow and generates a membrane like this for some regimes in the cell-like domain. And you can see we have plotted here the size in green and the mass, and the minus entropy, which is the information. And you can see that as it evolves, we measured that in this from the computer simulation, and you can see how the system, these equations, as the systems begin to replicate, how the system replicates and emulates the natural cell division cycle. In fact, we can show, we can dig into the values of the parameters in such a way that we do see that our system produces something which is a realization of this abstract form of the cell division cycle. So all of this is very nice, it's very interesting, and so on and so forth, but what the hell do you do with it? Well, the first thing you learn from this is that perhaps, since this is biochemistry agnostic, that is to say, the equations do not talk about biochemistry anywhere, they are just substances U, V, and B, and C. So this is telling us, these solutions are telling us that there are other conditions which give rise to the unification, other substances that give or other simpler models that can actually give you the properties of a living system without having to rely on biochemistry. In fact, it's quite simple what you see here. So then you say, you cite our friend Picasso, who said, everything you can imagine is real. Well, it depends on how many bottles of wine you drink, of course, but also it is real if you actually do it based on the scientific method. And then you can actually begin to ask the question that since our mathematical formulation is agnostic to biochemistry, so could we then use the principles of physics, chemistry, and engineering to design and construct systems that do all of this, capable of executing the functions of life? I have spent a lot of time discussing this, and not because I wanted to spend particular time in the theory, but because it is very basic that you understand where I'm coming from and where the inspiration comes and why. And I feel that I am playing at home, so to speak, talking at CAB, and I wanted to also show the roots of part of the work. The roots of part of the work is, of course, work that Federico, Felipe Lesmes, David Hackberry, myself did in the early, late 90s, actually, early when we were studying galaxies and all that, and then we came upon a simplified form of this, which we didn't understand, or I at least didn't understand it. I understood it much later. And then what I wanted to bring this to the fore. And then after this transparency, we say, well, all of this you probably can do. You can probably build a system that does that. But how? How do you actually put a chemically operated artificial living system? By artificial means that you're doing it in the laboratory. It's not artificial life like the one you do in a computer. No. In a wet lab, okay? But artificial, totally made by us, by human beings, which is capable of boot up like a computer autonomously from a state of high entropy. So from a homogeneous mixture, for example, you will encounter right away that unless you concentrate the materials in somewhere in a region of space and time, you will have to deal with something which is called the arithmetic demon, which I will not discuss here today, but which has very illustrious roots. In particular, an extraordinary chemist from Barcelona, Felix Serratosa, wrote a lot about this. And I recommend that you look at his work, tip of the hat to Felix. And how can you overcome the arithmetic demon? Well, the way to overcome the arithmetic demon is perhaps to generate some form of encapsulation where you keep the concentrations inside a particular region of space-time. But what is the set of initial boundary conditions? Well, something that actually is capable of spontaneously, so to speak, spontaneously generates an encapsulation of some kind. So can you do this autopoietically? The system itself generates this. We call these systems bichos, which, as most of you know, in Spanish is bugs. In English is bugs, but this is Spanish. And we put the two S's at the end for some reason. And bichos stands for biologically inspired, chemically operated synthetic systems. Okay. So, but how does it self-assemble? How does it self-reproduce? How can one program this? And does it communicate with others? And how can it adapt? Does it evolve? All these questions begin to pop into mind. Well, in the mid 20, around 2013, 2014, a postdoc of myself, we began to, I was already aware of how all of this worked. Alberto Pérez Municipality had already understood with me, we had understood these equations, how they worked and so on and so forth in details for the connection with the life cycle and so on and so forth. And we started looking for making polymersomes, that is to say, like liposomes, but using polymers, because polymers you can do artificially fairly easily. Lipids is more complicated and it's biochemistry, and I wanted to stay away from that. And we started putting inside, we started making them in a very manual way, using macrofluidics or using other methods, which were totally engineering methods. But we started putting, coupling the Belousov-Zhabotinsky reaction, which is an oscillatory chemical reaction. Unfortunately, I don't have time to discuss this in more detail today. But we started putting the BC reaction into polymersomes and see if the BC reaction actually changed or affected the polymersome in some way. The idea was, will it actually make it shrink or make it grow and then divide, and so on and so forth. So we started doing this test. But why, you may ask, the Belousov-Zhabotinsky reaction? Well, the answer to that question, which is a great question, is because the solutions to the three, because the equations that are wrote for you, down for you earlier, the three equations represent a model for the Belousov-Zhabotinsky chemistry. The model is called the Brusselator. And therefore, with the three equations, we had a clear indication that we could have some chemistry, that we could make it in reality in the laboratory, which could behave in the right way if we knew how to do it. Okay, so this is the connection with the BC reaction, is realizing that this kinetics actually represents, is used to represent the BC reaction. And that was an epiphany. So then we said, can the BC reaction compute? And then, of course, other persons and myself were working in the lab, and we actually put together the most basic chemical computer you can build, which is a chemical Turing machine, which eventually we patented and published. And now we are developing it into a chemical computer. But the Belousov-Zhabotinsky reaction offered, therefore, a representation of the equations. Therefore, it includes something about metabolism, about self-replication in some way, if we put the right boundary conditions, and maybe some adaptation. And definitely, it would also help with the control of the chemistry. So this is what Teresa and I saw. You will see it's published in communications chemistry. No, chemical communications, sorry. Chemical communications. I have an error there, not communications chemistry, which didn't exist then. This is in chemical communications, and this is what we saw. You see, nothing happens there. at the beginning, but then it decreased. Something happened inside, and then it grew again. So that we saw in the laboratory. Of course, the system, the polymerosomes, the containers were put in by hand, the products were bought outside, Sigma Aldrich and all that. So that gave us a very big boost in adrenaline, because it told us if we were able to make vesicles from a homogeneous system and do it with the chemistry, then we might be able to actually start a system like what we want to do. And lo and behold, we went ahead, we studied with a postdoc of myself, Jan Szymanski and myself, we began to study the system. And when we rediscovered something that people had discovered a few years earlier, which is that if you use raft polymerization, a form of polymerization, which is called raft and stands for reversible addition, fragmentation, chain transfer, then you generate polymer chains, and you will see why polymer chains, which are actually very uniform, instead of having the degree of polymerization being very different in the various chains that are produced in the synthesis. This has the ability to produce a monodispersed polymer. So this is very interesting. And we began to do experiments. Jan and I began to do experiments which were published in 2016. And we began to using a fairly simple system, which I will describe for you quickly. And we began to see in the microscope, we put two restrictions on the way our system had to work. One is it had to work in water, that we were the first ones to do PISA in water. And not only it had to work in water, but additionally, the system working in water would have to be larger than 1 micron. 1 micron is a critical scale, which we can talk about some other day, if you wish, but it had to be larger than 1 micron, because then we could follow the evolution of the system in the optical microscope. And you can see here that this little thing began to grow. It generated out of a blob of polymer, synthesized this. The start of this was a homogeneous system. It generated a little vesicle here, and the vesicle a little later had grown from something like 6 micrometers in diameter to about 10 here, excuse me, from about 20 here to about 26 or something like that. Okay, so this was great. And the chemistry is described here. The process as it happens is described here. You start with what you call a chain transfer agent. You start with which is attached to a hydrophilic chain, in this case, polyethylene glycol. And this is your starting point. And then you put a monomer in solution. And then in this case with photochemistry, and I hope this, I don't know very well if I'll be able to use this. Yes. So the polymerization begins to attach the monomer to the end of to where the chain transfer agent was, begins to touch the monomer, and the monomer makes when it polymerizes, makes a hydrophobic, PEG is hydrophilic, makes a hydrophobic chain, and then you make an amphiphile, a molecule which is has two parts, a part which likes water, so it's soluble in water, and a part which doesn't like water, which is a hydrophobic. These, as you increase the ratio, or you change the ratio of the length, the dP, the degree of polymerization of the hydrophilic part to the hydrophobic part, as that changes with the polymerization, the system first makes micelles, then it makes tubes, actually worms, they are closed, and then they become vesicles. And that is the beginning. So in these experiments, we put together, we brought into the chemistry, we made a homogeneous homogeneous mixture, and then we put these two as a single molecule that we synthesized, and that was one of the ingredients. Today, if you look at a paper in Central Science that came out last August, some peers and myself, we actually can start with simpler components and actually using a combination of click and PISA chemistry, you can actually make this even simpler, and we're very happy about this. So this is called PISA, this polymerization induced self-assembly process. It's done in a laboratory with very cheap, simple components. The experimental, what becomes expensive is not, well, this is not very cheap, but it is cheap compared with microscopes and so on. Then we have developed our own 3D printer instead of using cuvettes, which are expensive and so on. So we do 3D printing. Then we do experiments where we thermostat the system and so on and so forth. And the way the procedure goes is the following. We start with the chemistry in a vial. This is for this. This is from the original paper where we reported some of this behavior that I'm going to be showing to you in the next 2 or 3 minutes. And you start with the system which is totally homogeneous. You shine light on the system, and the chemistry is such that the system begins to phase separate, and the phase separation, it produces objects like what you see here. And those objects are active objects. That's very important. There is a combination between the physics and the chemistry, and the combination is that occurs is a combination that involves the properties of the say the properties of the vesicle, how it grows, and the energy in the surface of the vesicle, and how it evolves, and the generation of the effects that take place. And this is described, of course, by a set of equations which are called the Rayleigh-Placet equations, describe how the system behaves. What it tells you in our chemistry is that the system will grow, collapse, grow, collapse, grow, collapse, grow, collapse. During that process, and this is predicted by the hydrodynamics. And what you do see when you take this system and you take a sample from this part and you put it in the microscope, as I showed the microscope I showed to you earlier, you get systems that out of a homogeneous mixture, they begin to develop vesicles like this. They grow in size. You can see the curve here. They will collapse. They grow in size again. We call this phoenix. And you can see with your own eyes that around where these guys collapse, the population increases. And the population increases, you can follow the population increase, you can count. We know how to count with a microscope. And you can see that the population follows a Lotka-Volterra trend. This is very important because it means that something is competing with something. Okay, and this process of growth and collapse can be, as you can see, is a form of cell division cycle. Okay, so and you can see better in this transparent, in this other image. You can see how these boogers replicate. I know that I'm very close to the time, and I want to ask Marta or Victor if I can continue for about 5 more minutes.
Sure, go ahead.
Thank you. So you can see how the system grows, the number of vesicles grows. So there is a self-reproduction there. The self-reproduction, we understand exactly how it happens. And this is something we're writing right now. And what essentially happens is that the system micelles, and then it grows. Then there are some processes inside which actually generate some places where there is drops of water that form inside, and of course there are chemicals in there. Then there are osmotic pressure effects. Then there is relocation of molecules of amphiphils inside, and other molecules inside a double a vesicle with 2 walls. And then you see how there is some form of stress in the membrane, and the system here begins to grow. As I said, the membrane becomes thinner, the system grows. This is described by the Rayleigh-Placet equation, and the system develops defects on the membrane, and on the defects on the membrane, the system collapses, just like a bubble in your in water when you change the osmotic pressures and so on and so forth. That's a physical chemical process. And the system, when it collapses, spits out a lot of junk, including non-reacted molecules, and then it finds itself in a medium which contains food. And then what happens is that they grow, they put out all of these seeds, which are like spores, and the system begins to grow daughters, and then that's how the population actually increases. So this is what we see. This has become the cover of a number of journals, and we also can do the same thing with the BC reaction. We can do a similar system. That was a cover that Victor was mentioning. In Angeguante, there is also, we see other systems, and I don't know, we get invited to get covers. So it's quite interesting how that happens. So to end, I would like to mention something which is, we became very interested in something which is trying to see if there was competition. And if there was competition, was the competition such that it would satisfy one of the prerequisites for evolution that Darwin described in chapter 4 of his Origin of Species, the struggle for life. So the struggle for life is seen, essentially says that if you have two species in a single niche, in a niche where they share a at least one particular ingredient for food or for their own proliferation, then if one of them has a little advantage, then the other one will go extinct. And this is something which is very important in understanding the evolution of finches in the Galapagos Islands and so on, and it gave rise to a very long and beautiful intellectual description and phenomenological description of how finches evolve in size and the beaks and so on and so forth, which was preceded by some work by a person called Gauss, who noticed that when you took two species of Paramecia and you put them in this before the study on Darwin finches, two species of Paramecia, you put them together and they shared one particular ingredient in their food, then they followed very special paths. So one Paramecium, for example, called Paramecium aurelia, grew in numbers, and the Paramecium caudatum also grew in number. When you grew them to, this is if you grew them separately, but if you grew them together, one of them, in this case, Paramecium aurelia, grew better because he had a little advantage over the other ones. So can we build a mimic of that? And we have built a mimic of that. We have taken vesicles and we put, again, done by PISA, we put in one of them, we made a population of vesicles, and then that population of vesicles we separated into two populations, and then we doped one with zinc and the other one we left the original ones, and it turns out that they follow exactly the same pathways. And this manuscript is right on my desk right now, and it's going to be submitted during this weekend. We have also gone further, and we have developed and are developing adaptive chemistry. If you look at this paper in Central Science 2021, Sam Pierce and myself, you will see how we don't need to put the raft agent. We can start with copper, alkyne. I always forget the biochemistry, the chemical names of this thing, alkyne acide complex. You put or catalyst, you put this here, then you can actually begin to do the same experiments with the same type of behaviors that we get with a macro raft agent put there. So this is even simpler. And it, in addition, has two loops, and the loops work exactly as when you have an adaptive chemistry. And we're very excited about that because it's opening new doors for our bichos. So we do have I'm finishing now. We have been able to, starting from life, getting inspiration about what life is and so on, and thinking of equations that could describe living systems or could describe properties of living systems. We have gone into, we have been guided by the equations into going into generating systems which actually behave like what you saw in this picture. And if you do it with BC, you actually have a different type of replication. I didn't describe that. It's published in Advanced Materials. And then, so we have a very simple form of life, which is like the filter paper pot of coffee. Okay, this is what we have. It's very simple, and this is like the real the pot of coffee you probably have in your office, which is much more complicated. That's the same thing, coffee, and this the two do the same, but this is much simpler than this. So this is like the type of model we have. So we set out to mimic simple natural life using an outside-in approach, that is to say, starting from the top, the properties at the top, and going down to the details, and avoiding biochemistry. We're able to implement the basic properties of life with chemistry, but without using biochemistry. Starting with a homogeneous mixture of small molecules, one can boot up interesting autonomous systems which integrate above properties. And we're now working on the complete integration of adaptation, the complete integration, including full implementation of adaptation and information handling. This has been done with the help from many people, but in particular, those people you see here, Anders Albertsen, Vishnu Bastakoti. Sorry that I'm going to give the names: Gong Cheng, Thomas Draper, Marta Dueñas, Jean-Sebastian Agnon, Sudina Buragain, Sai Krishna Katla, Che Yulin, Alec Pauling, Sam Pérez, Alberto Pérez Muñuzuri, who is in Santiago. Marta used to work for Repsol. She's no longer with Repsol. Esther Poros Tarkali from Budapest, Keishan Ran, Teresa, that I mentioned her work, Rohit Srivastava, Eric Shabo, and Jan Szymanski. So all of that was done with their help, and that could not have been done if it not been from the help of Repsol and Harvard, the Harvard Origins of Life Initiative, and also the National Science Foundation, which actually funds the Center for Nanoscale Systems, where we can do all our work characterizations. And some other day, we can talk about how all of this perhaps goes into planetary systems and how it could what it could tell us about life in exoplanets. And I will shut up and I will stand for questions if there are, and it will just let Victor or whoever continue with leading. Thank you so much.
Thank you, Juan. Thank you very much for such an interesting talk, and a special thank you for making it understandable for the ones of us that are not working in this field. So time for questions. If you have questions, please raise your hand. If you don't have a micro, you can write it on the chat and I'll read it out loud. So go ahead, please. Let's see. There is one hand. Go ahead.
Hello, Juan. I'm very glad to hear that the 1947 generation is still active.
That's Josep Marie. Josep Maria.
I'm doing very nice work. My only question is that in the last slide you show, I saw some metals, and as you know, I like metals very much. You can repeat these experiments without them, or they are very important for the kind of reactions you are studying?
They are fundamental.
Okay.
They are fundamental. Ricardo, good to see you now. Okay, the metals are fundamental. One of them is ruthenium. So you saw ruthenium, you saw zinc, and you also saw copper.
Copper, yes.
Okay? Copper is fundamental for the click reaction. For the PISA reaction, what is fundamental is ruthenium or zinc. When you have ruthenium absorbs in blue light, zinc absorbs green light. So you can play with illuminations to actually make the systems adapt and so on, be more effective in the chemistry and therefore win competitions which is what actually happens in the paper that I have, that the paper about the big, hang on a second, because somehow the communication failed for a bit. So we use light to actually favor one species or another, and so on and so forth. So metals are fundamental because they work as catalysts, and because they work as catalysts. You need catalysts.
Great.
Thank you, Ricardo.
Okay, so there is another question from Jacobo Aguirre. Please go ahead.
Jacobo, enséñame la cara si puedes.
Venga. ¿Se me ve?
Te veo perfectamente, aunque con el contraste detrás. Me alegro mucho de verte. Please go ahead.
Igualmente, Juan. Well, thank you very much for this extremely inspiring talk. And I have quite a general question regarding actually your last transparency, where you talk about that you're starting to work on information. How does your all these marvelous experiments fit in your mind with how information is fixed and is reproduced afterwards? You know, if you want, I'm talking about this RNA world or this information understanding of the beginning of life.
Yeah, that is a very wonderful question. Okay, and it will require, besides my usual verbose answers, I'll try to make it short. Okay, but it's a very wonderful and important question. So when you said the last slide you showed, I thought you meant this one. So let me go back to the beginning of the talk and pop this up. So we are in a laboratory. Of course, we're not in the origins of natural life or anything like that. Mm-hmm. We are in a laboratory, and the work that I described to you would correspond to things right here, or probably even before that, okay? But it's work that happens in my lab. And of course, I am choosing, or we are choosing the substances, we're choosing the scenario. We have a lot of information already when we do our experiments. That information leads to our choice of molecules. And the molecules we choose themselves have some information. It's called conformational information. Okay, that conformational information makes the molecule go to one stable state or another stable state. Depending on the stable state where you are, you have more information or less information, conformational information. The information is in the shape of the molecules, and it's also, and the shape of the molecules, of course, is a consequence of the atoms and how they are laid in the molecule. When the molecules combine in a chemical reaction and they react, you are actually doing a computation. So the information that is in the molecules is input into the chemical reaction. The chemical reaction transforms the information that was originally input in the molecules that reacted and generates a product, an output. A computation is a process by which information is input, mechanically transformed, which means that always the same inputs always lead to the same outputs, and it's transformed into an output that can be used for a different purpose than what it was here. Okay, so that is done in a chemical reaction. Different chemical reactions have different properties, and information can be very simple or can be more complicated, and so on and so forth. There is a very beautiful hierarchy of information. It's called the Chomsky Hierarchy, that tells you, and we have generalized that Chomsky hierarchy of languages and automata to chemicals and chemical reactions. But you start with the simplest form of information that you can imagine is the information in the forms and the shapes of the molecules at the very beginning. Right. Okay? And then that information, when the reaction takes place, generates, it's called conformational information, generates another type of information, which is configurational. It gives the configurations. So you're using free energy to transform it into information. And remember that the free energy contains a term which is T times S. S is entropy, and minus entropy is information. So the chemical reaction actually is transforming the information, into configurational information. That is the type of information we're using here. The information, for reasons which of course we do not understand yet, but we will, and I will say why, that information eventually became some digital information in the sense of bases, like the bases in RNA and DNA.
Mhm.
And if you look at DNA and RNA, and you look at some regions of RNA, you can actually see that they contain, they are written in some language, in some of these languages of the Chomsky hierarchy in terms of bases. So what this is telling us is that there is an evolution in the nature of information from conformational information to configurational information to genetic information. How this happened is, of course, a mystery, but there is a pathway to it through chemistry. It's quite clear that there is such a pathway. So if you want to learn more about this, you could read the work of Robert Shapiro, okay, who was at the, so you could read this book, or you could read a Scientific American paper by him, which was published in 2007, and where there is a description of many of these things, which some of the things we've been discussing, not the chemistry, not the details. Okay, thank you for the question. And if you want to ask more, please shoot me an email.
Thanks a lot. Thanks for the talk.
Thank you.
Okay, so I don't see any more questions.
I have one question, because I don't know where to raise the hand in my screen.
Okay, go ahead.
Okay, well, thank you, Juan, again, for your talk. Is every time you explain this is a new way of looking at the original lab, I think, and looking at the original biochemistry, and I think it's very, very inspiring. And I have a question, because I see in your vesicles that there are certain heterogeneities, irregularities, some places where you have accumulation of reagents, something like that. Don't you think that this can be a kind of extra compartmentalization? Don't you think that these places could somehow play in a different role? Or another way, can you modify the experiment so that you somehow add an extra degree of complexity and creating new spaces where the chemistry enter in a different stage? I don't know, in a different I would say scale. Would it that possible?
That's a very nice question. And that hooks up with a question that Ricardo asked a little bit about, which is the presence of metals. This is what happens in the Phoenix type of replication, okay, and chemotaxis. It also has chemotaxis. I didn't discuss that in the talk, not enough time, and you may not. I wanted to allow for people who may not be interested in these things, okay. So, and they just want to hear something and not many details. But there are many things that do happen as the vesicle grows. There are defects which are unavoidable. And they're unavoidable, and they produce defects in the surface. And those defects are the consequence of quantum fluctuations, thermal fluctuations, all kinds of fluctuations which are always present, and you will never be able to shut up. Okay, so it is the presence of those defects that actually generates pores in the system and makes the system collapse. The system that you see here only has ruthenium. The ruthenium, when we generalize this to the BC reaction, the ruthenium can also be the catalyst that the BC reaction does, okay? But we started, as I mentioned in the talk, we started thinking about how do you put two, I didn't say this explicitly, but what we started thinking about was how do you put two loops into the system? How do you bring two loops into the system? and how do you connect them in such a way that the system is adaptive? What we found is that we need to do click chemistry. This is explained. So you have the click chemistry and you have PISA. The click chemistry occurs here, this part here, and the PISA occurs here. Okay, and then this is the piece, the amplifier synthesis, and this is the self-assembly takes place here, and here is the vesicles. But what you do see here, and I didn't have a chance to explain this clearly, and I'm not going to explain it now. What we do see here is that in order to add more complexity to the system, a very clean route of adding more complexity is to put more catalysts inside. And the cheapest, not monetarily only, the cheapest catalysts that are, or the easiest catalysts to involve, are metals. So that's why we went to click chemistry with copper. Okay, so yes, the answer to your question is yes, you can increase the simplicity, and at the same time, as in this case, increase the simplicity and at the same time increase the complexity of the resulting system. It's amazing, but you can do it.
Thank you, Juan.
You're very welcome, Victor. Thank you for your question also.
I have a question. I don't know.
Yes, go ahead, please.
Yes, I am in line. Yeah.
Hey, Jose Maria.
How are you?
Happy to see you.
Yeah, happy. Me too. In the case of these polymersomes and in all the capsulated systems, sure you have some idea of the effect of the selectivity of the membrane to species from the surroundings, to acid and so on, not only to the water, to the chemosmotic pressure. Probably, I don't know in these publications, but do you have some results in relation, for example, simply to proton permeability of the membrane?
Yes, yes, we have a yes.
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 Will you say something about this, please?
Yes, of course. First of all, let me give you a reference that you might find useful for your question. Okay, this is a paper that came out in Soft Matter.
Yes.
Where we use PISA with pH, with the pH oscillator, not PISA. Okay, so we do understand a lot about the proton affinity on and the membrane, what it is permeable to, what it is selective for, which kind of molecules it is selective. We have a lot of information about that, and we have characterized, not exhaustively, because that requires a large team of people, and our resources are limited. Okay.
Yes. Yes.
So, but it requires a large team of people to do actual really meaningful characterization of the surface. But we do have a lot of information on selectivity with respect to molecules, external molecules, selectivity also in terms of whether there is I cannot think of the technical term now in English, some agglomeration of chemicals, competition of chemicals within the membrane. That's also very important. Okay, so we do have information about that, and we have spectroscopic studies and SAC studies saying something about this. So the answer is yes, but more needs to be done.
Thank you. Thank you.
You're welcome. I know what you're asking. Okay, take a look at this paper.
Yes, sure.
Okay.
But you mean with pH oscillator, that is the change of pH in the surrounding, no? Probably.
No.
That is the pH inside.
This is, instead of the BC reaction changes, the oscillations in the BC reaction changes the redox potential.
Yes.
Okay. In the change in these oscillations, there is the production of radicals.
Yeah.
There is an inhibitor, etc. So you have cycles of inhibition and promotion. And those are the cycles in the BC reaction. But in this paper, we do not use the BC reaction. We use a pH oscillator, which is an oscillator a chemical reaction that autopoietically, if you wish, by itself, actually oscillates in pH. It's a very complicated reaction. And that generates vesicles also. If you are interested in these pH oscillators, you may also want to look at the cover. It came out a week ago of ACS Omega, where we have a Turing machine based on a pH oscillator, which can now be used to make information controlled vesicles like these ones.
Yes.
But using a pH oscillator instead of using the BC reaction. It's an amazing thing.
Yeah, sure. I read it, sure. Thank you.
You're very welcome, and it's very good to see you also.
Yes, I am very happy to see you.
Yes, me too. I'm thinking of our common friends,
Yes.
some of which are not with us anymore.
Yes.
Okay, so are there more questions?
Yes, can I ask a question, Juan?
Yes.
Can you hear me? Hello, this is David. It's good to see you.
Hey!
Very good to see you. And first I wanted to say hello. I haven't seen you in a while. Un abrazo muy fuerte. And I had a question for you. Okay.
Yep.
Of course, your systems are open, non-equilibrium systems, of course. And one way to characterize, as you know, open non-equilibrium systems with non-equilibrium thermodynamics is to consider the dissipation, the entropy production, and also the flux of entropy between your system and the outside world. Now, I was wondering, since you have these nice model equations that you've been studying for many years with Alberto Muñozuri, you would actually be able to calculate within the context of your model the entropy production and also the flux of entropy between your system and the outside world. And I think it would be interesting, if you haven't done this, then to complement one of the plots I saw in your talk at the very beginning. You had a very interesting plot there, which was showing the growth of both mass and information in a cell versus time, which looks sort of like a ratchet. Things would grow and they come down, they grow and they come down. I think it would be interesting. You could track also the dissipation and the flux of entropy in your non-equilibrium systems and complement that graph with how the dissipation and the flux is perhaps growing and decreasing through these cell replication and division cycles.
The answer to that is we haven't done it, but I have in other work, I understand how the dissipation of the entropy production is actually related to the free energy change in the system. And this is a very important point in computation, in chemical computation. If you look at a paper which is in Frontiers of Chemistry, a review paper on on the BC reaction applied to chemical computation. The paper came out last year, I think 2021, sometime in 2021, February or March or April 2021. It's Marta Dueñas and myself. It's a review on chemical computation. You will see that there in chemical computation, I'll come back to your question in a few seconds. I don't want to keep people there. If you, in that paper, you will see that during a chemical computation, there is something very important that takes place. And that is that for any word that is recognized by an automaton, the generation of entropy flux is equal to -1 divided by the absolute temperature times the change in the free energy, the Gibbs free energy in the system. And it is the same, that equation is the same for all words that are accepted by a chemical computer. So, having said that, it is clear to me that the graph that you saw, to me and to many people, not only me, of course, to anybody who's thought about this problem, and there are many people who have thought about this problem, when you see graphs like this, you're actually doing a computation of some kind. Okay, and you have here the change in free energy, you have here the change in entropy generation and so on in this graph, which is of course what the model that we now have produces to replicate this. So yes, the answer is that this can be seen as a computation, and I know that computations satisfy this beautiful theorem. And you can find information in the Frontiers in Chemistry paper. And we can talk about it if you wish, of course, and maybe discuss it with a JAS or with Alberto or something, of course. Yes.
Okay, thank you. Thank you very much, Juan. Thank you.
Thank you. Thank you. And thank you for all the work we've done together over the years also.
Oh yeah, it was a lot of fun, right? That's the whole idea, to have some fun and to learn things.
Still is. It wasn't.
Still is, yes.
Still is. Very important. Yes.
Yeah.
Yeah.
Okay.
Any more questions? No, I can't see any questions in the chat either, so I think we can leave it here. So I want to thank you again, Juan, for being with us today, and we hope to see you maybe in person in the future. So thanks, and thank you everybody for following the seminars. Thanks.
Thank you very much for the invitation, Victor and Comisión de Seminarios and CAB. I wanted to dedicate this talk to Mamen's memory. And I would also like to thank everybody at CAB for paying attention today and for looking at the work we do and for your questions. And if you have any additional questions or you would like anything you need, please do send me an email. You know, my email is easily found. Okay, so take care and have a wonderful day. Thank you so much to everybody. Good to see everybody.
Thank you, Juan. Bye, everybody.
Muchas gracias, Juan.
Ay, muchísimas gracias.
Encantado. Gracias, Álvaro. Gracias a todos. Gracias.